I'm Tyler Mann, and my presentation is on carbon nanotubes and carbon nanotubes in protein detection assays or tests. So to begin, what are carbon nanotubes? Carbon nanotubes are tubes composed of purely carbon atoms in hexagon lattice that close together to form a ring, and they can be single-walled or multi-walled. Single-walled nanotube is displayed on the right there. A single-walled nanotube is usually about 1 to 2 nanometers in diameter, where a multi-walled nanotube is 10 to 70 nanometers in diameter. And a multi-walled tube is just multiple single-walled tubes inside of each other to form shells. So they have some pretty unique properties. Firstly, their size, being only a few nanometers wide and usually a couple hundred nanometers long, they're able to interact with a large variety of small molecules at a scale relative to these molecules. They have a really high strength because of the hexagon lattice and their ring shape. They are extremely strong, stronger than a lot of metals or other materials. They have extremely unique fluorescence and conductivity, and I'll get to those later, but it helps them to be useful in protein detection tests because they are easily detectable. So what are protein detection assays or protein detection tests? They are useful tools for everyday life. They'll test for a specific protein in a sample, and testing for a specific protein is helpful because the body expresses proteins in response to unique situations. For example, if you're sick, your body will express a certain protein, and if you have a test that is able to detect these proteins, you can determine if someone is sick or not with confirmation. So you can use this on all sorts of things like cancer, disease, infection, medication, interactions, etc. A few examples are on the right here with COVID-19 tests, pregnancy tests, and drug tests. But a lot of the current objectives of these tests are to become more specific, more sensitive, and faster. So why should we use carbon nanotubes for these tests? Carbon nanotubes are good candidates because they can be functionalized or complexed with antibodies, enzymes, or DNA to detect other biological molecules in a solution. In my examples, I'll be using antibodies, and they're able to effectively interact with biological molecules because they have similar size, because they are so small, and due to the fact that they are only made of carbon atoms, they are non-toxic to biological molecules. They have an extremely high surface area due to their shape, and this means you have a lot of area to put whatever biological molecule you intend to use on the outside of the tube. They also have distinct signal changes when their structure changes. For example, if your antibody binds with a protein of interest, you have an immediate change in the structure of the carbon nanotube, and that can be detected to detect the level of proteins. So how does it work? Like I said, I'll be using antibodies in my example. You complex them to the outside of the nanotube. They're shown here in blue. You then introduce this complex to a solution where you want to test for protein levels, and the proteins are shown here in pink. If the proteins of interest are present, they will complex with the antibodies. And when this happens, the overall structure of the nanotube will change almost instantly, and you can detect these changes in electrical signals or fluorescent signals. And one of the more unique properties of carbon nanotubes is that their fluorescence is just outside of the wavelengths of a lot of natural biological molecules. So this makes them really easy to detect inside biological solutions because you don't have other interference from the other molecules in the solution. So why does all this matter? If you can get a sensitive test, you are less likely to miss a diagnosis, and you need smaller samples from a patient. So you would only need a drop of blood instead of a whole vial of blood, for example. They are extremely small. You have handheld testing and at-home testing because of the fact that carbon nanotubes are so small, you can put them into virtually any test. And if you can get handheld or at-home testing, this makes them more accessible to everybody, so everybody can have tests, and it can make them faster. Large contact area means detection can happen quickly because you have so many antibodies on the side of the nanotube and the structure changes almost instantly. You can have results be read almost instantly in some cases. So what is the future for carbon nanotubes? So there's a couple possible advancements that have been done. You can have injectable testing where your complex of nanotube is injected directly into the body. and this would allow for in-body monitoring of protein levels. And this can be done for days or weeks at a time, and that's thanks to the fact that carbon nanotubes are extremely strong and long-lasting due to their shape, so you can do monitoring for a long time. You can also do multi-complexation, where you attach multiple antibodies that are specific to different proteins onto the same nanotube and introduce that to a solution and be able to detect multiple proteins in a solution at the same time. The biggest holdup right now is production. They are difficult and expensive to produce at a large scale, and that's really the problem right now for why they're not getting a lot of use. But they also have a lot of other cool biological uses. For example, drug delivery, where you can put medication inside of the tube itself and then use that to deliver your drug in the patient. They are also used in tissue engineering, where they can be used as a scaffold for the tissue to build itself upon since they are so small. So, what are the takeaways? They have been extremely successful in protein detection tests. They've proven that they can detect proteins highly sensitive and be effective, and they could significantly improve our testing capabilities if a little more research and a little more advancement is done. And they are advanceable, which is good news. So further advancements could see them become more versatile in the medical field than they are right now. But the big problem right now is production limitations, so we need to find easier, faster, more affordable methods to produce these carbon nanotubes before we can use them in a day-to-day basis. Thank you.